Good morning, my dear students. Today we are going to take up a very new important topic in botany, that is what is called as sexual reproduction in plants. On the other day we were discussing about vegetative propagation, how the plants are vegetatively propagating. Today the discussion is going to be on sexual reproduction. Now, you know, for the sexual reproduction, the plant needs the flowers. You get male sex organ and a female sex organ inside the flowers. And this takes place, the male sex organ is called as androecium and the female sex organ is called as gynoecium. These two sex organs, namely the androecium and gynoecium, are enclosed by two non-essential organs, namely calyx and corolla. We call them as non-essential organs not because they are not essential for the flowers, but they don't do or they don't involve themselves directly in the function of the reproduction. Okay. So, it is for that purpose we call them as non-essential organs. Now, so a flower contains four important structures, namely calyx, corolla, androecium, and gynoecium. In this, the androecium is the male sex organ and gynoecium is the female sex organ. These two are the coverings. Now, what is a sexual reproduction? It's a biological process involving the fusion of the gametes of the two sexes, namely the male and female, leading to the formation of a fruit and seed. Flowers are morphological and embryological marvels on the site of sexual reproduction. So I told you the flowers are the sexual reproducing organ in the plants. In flowering plants it involves the two processes, namely meiosis and fertilization. So you may be wondering what is this meiosis and fertilization. See, each and every organism in this world is composed of chromosomes. So in a plant you have got the male sex organ and female sex organ. Now a combination of these two only gives rise to an organism. Now in this male sex organ the chromosome number will be 2n. And in the female sex organ also 2n. Now if they combine together, then the organism will become 4n in nature. So what happens? A reduction division takes place in the male sex organ as well as in the female sex organ, leading to n number of chromosomes in the male gamete and female gamete. Now when they fuse together, you get once again the organism formed with 2n number. It may be a male or it may be a female. So now this process is what is called as reduction division. Technically it is called as meiosis, and this stage is called as fusion. Fusion. Or it is also sometimes called as syngamy. Syngamy is another term used. What is the meaning of the word syngamy? Gamy means gamete, syn means a fusion, the fusion of the gamete, that is the meaning of that word. So this is the reduction division taking place at the time of the formation of the gamete, and this leads to the fusion of the gamete. This is taking place for the restoration of the or for maintenance of the chromosome uniformly generation after generation. Okay. So that's what I have written here as a meiosis and fertilization. Syngamy is also called as fertilization. Now, the beautiful diagrammatic representation of a flower. As I was telling you, this is the calyx. This portion is called, this is a calyx. And this is a corolla. This is a corolla. Okay. And these are all the male sex organs. This is the female sex organ. Okay. Now the pollen grain enters into the female sex organ for fertilizing the ovule to become the seed after some time. Okay. Now here you see they have labeled this as a sepal, but I have written it as a calyx. So a collection of a sepal is called as a calyx. Similarly, a collection of a petal is called as a corolla. Many petals form a corolla and many sepals form a calyx. Many stamens form the androecium. Many carpels form the gynoecium. So this is the structure of a flower. Now, how the reproduction is taking place? The same one just now I explained with the help of another diagram. See, it takes place. Egg and the sperms are produced. In the case of the plant, is a uniform diagram for representing anything for that matter. It could be for an algae, fungi, or a plant or animal, whatever it is. So it's a generalized diagram. So they have used general terminologies here. Here it may result, egg means ovule, and the sperm means a pollen grain. Fertilization results in the formation of the zygote or syngamy, from which embryo develops, and then you get the sporophyte. Okay? Then meiosis results in the formation of the gametophyte. So this is how the life cycle is taking place, and this is a common representation for any organism in this world. They have written as a flowering plant life cycle, but this is the same thing for even human being, plant, animal, for everything. The same procedure is taking place. This beautiful picture tells you how the reproduction is taking place in the plants. I just start from here. As I was explaining to you, the flower is the reproducing organ. These are the petals, sepals, etc. is over. These are the stamens. These are the stamens. And this is the gynoecium having the style and stigma. Okay. Now they have just shown it very clearly. This is the male sex organ. This is the female sex organ. Male sex organ, TS of the anther they have shown, and this is the ovary. And a fusion of that is resulting in the formation of the ovule. So our complete overall diagram this is. So each one we are going to study in a very detailed way in the slides that you are going to see. So I just skip this slide because each and everything is going to be depicted to you in a very detailed way in the slides that are going to follow us. Okay, fine. So a bisexual flower consists of a calyx, a corolla, androecium and gynoecium. Calyx and corolla are not essential organs. Androecium is the male sex organ and gynoecium is the female sex organ. It is divided into 4 topics. What I am going to discuss is broadly for my convenience. I divide this into 4 steps or 4 areas. The first one is what is called as a pre-fertilization. Before fertilization, what are the changes that are taking place in the plant? I am going to describe, and that is once again divided into 3 areas, namely microsporogenesis and megasporogenesis and pollination. Microsporogenesis means the formation of the pollen grain, and megasporogenesis means the formation of the egg, and pollination means the way in which the pollen grains are brought in contact with the egg. In fact, the way in which the pollen grain is brought over the stigmatic surface is called as a pollination, and after that, when the pollen grain is growing into a pollen tube and then reaching the egg, that area is called as a fertilization. That goes by another step. That's another area. So for microsporogenesis, megasporogenesis and pollination are the 3 steps involved in the pre-fertilization, and then that leads to the fertilization. Then you have got a post-fertilization, which is once again divided into 3 areas or 3 stages, and then finally apomixis and polyembryony. What is this? Whenever there is a rule in botany and zoology or anywhere, you get the exceptions also. There will be exceptions. Nothing will be there in the world without exception. So when a normal embryo is formed, abnormal embryos are also formed. This abnormality is of many types. So one or two now abnormals we are going to discuss. One is called as an apomixis. The other one is called as a polyembryony. The formation of many embryos within the seed is called as a polyembryony. We will discuss about that in a detailed way. Okay. So this is only a pictorial representation. Once again, a bisexual flower is containing the androecium and gynoecium. The androecium is containing the filament, connective, anther. Then the anther is containing the outer wall, epidermis, endothelium, and the middle layer, the tapetum. These are the different layers of the anther. Then the microsporangium is undergoing a meiosis to produce the pollen grain, which is containing the exine, intine, the plasma membrane, vegetative cell, and the generative cell. And as I have already discussed it to you, the pollen, the gamete will be containing only n number of chromosomes inside that. See, this is a anther. This is a structure of an androecium stamen. Now you see, it looks like this. What I am drawing is the filament, and then it is having what is known as an anther. This anther, filament and an anther. This anther, when you cut, it becomes a 4-lobed, as you are seeing it here. So it is a tetralocular, and you call it as what is known as each one is called as a microsporangium, and this microsporangium. Okay. This inside each microsporangium there will be pollen grains, and this is the whole structure of the antherium. When you cut the anther, this diagram is as the anther is cut in this way. Okay. See, when you cut the anther, it looks like this. All the detail, all of them are shown in a very detailed way. This is a vascular stand. These are the 4 microsporangia. Now inside each microsporangium you get the microspore mother cell. Popularly it is called as MMC. We call it as MMC. MMC means microspore mother cell. Okay, microspore mother cell is popularly called as MMC. The microspore mother cell will undergo a reduction division to produce the microspore. Microspore mother cell will undergo a meiosis to produce the microspore. Microspore is otherwise called as a pollen grain, but there is a little bit of difference between the microspore and the pollen grain, because when what is escaping out of an anther is called as a pollen grain, but when the pollen grain is coming out, it will not be in a single celled stage. Microspore is one celled, but sometimes the pollen grains are in 2-celled stage or a 3-celled stage, depending upon the plant, depending upon the situation. Sometimes you may be inside each anther, you may be getting some pollen grains with a 2-celled condition or 3-celled condition. So pollen grain is not an equivalent term to microspore. That you have to understand very clearly. Microspore is produced as a result of reduction division from the microspore mother cell. Then it becomes a pollen grain. When it is becoming a pollen grain, the term pollen grain in English is given to a microspore grown in the form of a pollen tube. Okay, then it will be called the microgametophyte. Sometimes we are using the word microgametophyte also. So this is the epidermis. Then this is stomium, means mouth. This is the way how it breaks, and then through the mouth all the pollen grains are coming. So it's called stomium area. Then middle layers. Then tapetum will be the. So these are the different areas. This is the tapetum. This is the tapetum. Okay. Now, so I told you that I am going to divide this into 4 areas. First one is a pre-fertilization. Stamen has a long and slender stalk and terminal bilobed anther. The proximal end of the filament attached to the either thalamus or petal. The number and length of the stamens are variable in flowers of different species. Perhaps in this transparency, you may be having a doubt of what is called as bilobed anther. Bilobed anther. So, what is a bilobed anther? I just explained to you. So, when the anther is like this, the T is just now we showed, it is a tetralocular, or 4 microsporangia will be there. At the time of dehiscence, now what happens bythecus? These 2 anther microsporangia on this side and 2 on this side, they coalesce and then they become united, and then it becomes a one chamber. This becomes another chamber, and then the pollen grains here, they escape out through this stomium, and the pollen grains which are here, they mix it together and then they go out through this stomium. Okay, so that is why it's called a bilocular or tetralocular. It will be 4 chambered initially, but afterwards it becomes only 2 chambered because of the union of the 2 chambers which are on the same side, because it can open only on 2 openings. Only it in fact, I will be drawing one more diagram to explain that very clearly. Now this let's imagine this is a microsporangium number 1 and 2. So 1 and 2 will be there. 1 is here, 2 is on the other side. Similarly, 3 and 4. 3 is on this side and 4 is on that side. So 1 and 2 will coalesce, and then all the pollen grains will come out through this hole. Okay. Similarly, 3 and 4, it will be coming out through this hole. It is like that. Okay. So that is the meaning of that terminology. Bilobed anther. Okay. Now, a typical angiosperm anther is bilobed, with each lobe having 2 theca. 2 thecae. Theca means covering or chamber. Dithecous anther. Separated by a longitudinal groove, anther 4-sided tetragonal structure with 4 microsporangia, 2 in each lobe. Microsporangia develop further and become pollen sacs packed with pollen grains. Structure of the microsporangium. I think I have to explain to you one more very important one before I proceed to the next one. In the previous slide, we saw that the anthers are 4-lobed and then dithecous, etc. There is an exception for this. For example, in one, this flower that you have studied, you would have by now studied that the anther will be like this, and this is the filament. Now this is the condition where you will be getting only 2 lobes, and this is the best example for monothecous. Now by now I think you would have recognized that the name of the plant is Hibiscus rosa sinensis. Hibiscus rosa sinensis. In fact, this is what is called as a characteristic feature for the family Malvaceae. Malvaceae is characterized by many characters. The most important character of that one is what is called as anthers unilocular. Only 1 chamber will be there. It won't be, 2 theca. It is not a bitheca as we have studied in all the other plants. Okay. Now, so structure of the microsporangium. It appears as circular in transverse section. All these things we have already studied. It is surrounded by 4 wall layers, namely epidermis, endothelium, middle layers, and tapetum. Tapetum is a nourishing layer. Now, it supplies a food material for the plant, for the pollen grains which are developing there. So, pollen grains will be developing there. Whenever anything is developing inside an organism, it has to be supplied with a nutrition, nourishment. So it is the tapetum which is supplying the food for developing pollen grains. Outer three layers protect and they keep the and they help in dehiscence of the anther to release the pollen. Tapetum cells of the innermost layer have more than one nucleus with a dense cytoplasm. Just now I was telling you its function is nourishment. It nourishes the developing the pollen grains. In young anther sporogenous tissue, a group of compactly arranged homogeneous cells occupy the center of the microsporangium. As I was just now telling you, it is a microsporogenous tissue which is responsible for the production of the microspores. Now this sporogenous tissue is developing from the microspore mother cell. The microspore mother cell is continuously going on dividing to produce a sporogenous tissue, and then the microspore mother cell undergoes a reduction division final stage to produce 4 pollen grains. This is the, once again, this is the connective region. These are all the wall layers. This is the tapetum, and inside that you are able to see the microspores developing. The development of microspore is called as a microsporogenesis. You have to understand this term very clearly. Genesis means development. In biology, genesis means development. There is a subject called genetics. It is the study of the development. How a human being is developing, how a plant or animal is developing, it's called genetics. So genesis means development. Embryogenesis you would have studied in zoology, how a human embryo is developing. So genesis means development. Microsporogenesis means the development of microspore. Here the cells of a diploid sporogenous tissue, I was telling you, MMC, microspore mother cell, divide meiotically to form 4 haploid microspores. These microspores are arranged in a cluster of 4 microspore tetra. And once again, I think I need not to go into much details of that. It's once again a separate topic. These 4 microspores, they may be arranging in this form, or it may be arranged in this form. 1, 2, 3, 4 will be on other side. They will be arranging like this, or it may be arranging in this form. So different methods of arrangement of the 4 microspores are there. Okay, so it gets arranged, but whatever may be the type of arrangement, they separate out and then finally they go away as a single microspore. Finally they go. Then they mature, and then the intine, exine, everything develops, and they become the pollen grain. Several thousands of pollen grains are released with the dehiscence of anther through longitudinal grooves. I was explaining this to you. Pollen grain represents a male gametophyte. They are generally spherical in nature, 25 to 50 mu dia. I think I was explaining to you in another class what about mu. Mu is 1 by 1000 of a millimeter. 1 by 1000 of a millimeter. That's what it's called a mu. So its measurement is a 25 to 50 mu. You can just imagine. You can very easily imagine how small they are. They are powdery in nature. They are dusty in nature. Even the largest pollen grain, if you put the pollen grain one by the side of another, you can put 20 pollen grains within 1 millimeter. The 1 millimeter means the 20 pollen grains you can put. 20 into 50 will make into what's called as 100,000. So such small structures these pollen grains are. Okay. Exine outer hard layer is made up of sporopollen, one of the most resistant organic material, and it can withstand high temperatures and strong acids and alkali. It has prominent apertures, namely germ pores, through which the pollen grain germinates, and then it is forming the pollen tube. Pollen grains are preserved as fossils because of sporulin. Exine exhibits different patterns and designs. Intine and the inner wall is a thin continuous layer. It is made up of cellulose and pectin. This is the structure of a pollen grain I have given to you. See, beautiful, how nice it is. This is a pollen grain with the exine. So this is the exine. See, it is a very hard layer, and it is a sporophyllin. I have just now I told you, it is resistant to the heat, it is resistant to the acids, and it is resistant to so many things. That's how it is being preserved for many long time, for thousands and thousands of years. It is being preserved. When you take the petroleum from the earth, this petroleum will be containing a lot of pollen grains. Okay, and then by studying the pollen grains which are getting mixed with the petrol, you study about the type of the plants which are living about many thousands of years back. So the study of the fossil pollen grain is called, there is a separate paleontology. You have got a paleontology, the study of biological organisms. Then you have got a paleobotany and paleozoology. Then you have got a paleopalynology. What do you mean by paleopalynology? The study of the pollen grains which were living millions of years back. So the pollen grains, they have become very important tool, very important tool in the study of the evolution, because they live for many thousands of years because of their very strong exine nature, and then their capacity to tide over all the unfavorable climatic conditions. Fine. So now a single microspore has germinated into, it has come to the two-celled stage. Once it is becoming two-celled, you call one as a generative cell, a smaller one, and a large is a vegetative cell. The larger one, once again I repeat, the larger cell is called the vegetative cell, and then the smaller cell is the generative cell. Now the pollen tube is going to be formed, and then the generative cell is going to grow, and then finally it is going to bring about our fertilization. So this is, I think, mostly we have come to the end of one story. So in this story, one hero and heroine are involved. Our hero is the pollen grain and our heroine is the egg. So I am going to trace it in the form of two parts. The story about a hero is mostly over. I think a few more slides are there. See, this is how the pollen exine is breaking, and then the intine is growing, and then it is coming out in the form of the pollen tube, which is helping in the formation of fertilization. Plasma membrane surrounds the cytoplasm. Mature pollen grains contain two cells, vegetative and generative cell. Vegetative cell is a bigger with abundant food reserve and a larger irregularly shaped nucleus. Generative cell is a small and spindle shaped with a dense cytoplasm and a nucleus. In more than 60% of pollen grains, pollen shed at this two-celled stage. I was telling you, sometimes it is shed at the three-celled stage. In remaining species, pollen are shed at a three-celled stage when two male gametes are formed due to meiosis of a due to mitosis of a generative cell. On the shedding of a pollen, they land on the stigma. This is what is called as a pollination. This stage is called as a pollination. So, pollination, before they lose their viability, which is variable to cause the fertilization. Now, viability depends on temperature and humidity. They can be stored in liquid nitrogen, see, minus 196 degrees Celsius for several years. It can be used as a pollen bank, similar to seed banks. Viability is 30 minutes for rice and wheat. Very short viability period is very less. Now, before this 30 minutes, if the pollen grain is not able to germinate over the stigmatic surface, then perhaps it cannot germinate at all. So, before the viability period comes to an end, the pollen grain should find a place, it should land safely, and then it should be able to bring about the fertilization. Otherwise, it dies. Similarly, those of Rosaceae, Leguminosae, and Solanaceae maintain viability for months together. So, the viability period ranges as short as 30 minutes and as long as many months. So, there is a very broad range of the viability period. Pollen grains have, sorry, is a spell, I mean grammatical mistake. Pollen grains have both positive and negative effects. Positively, they are rich in nutrients. Pollen tablets and syrup are used as a food supplement in Western countries. Pollen consumption of athletes and race horses seem to improve their activities. So they are very rich in food. So that is why it's being used by energy-rich materials. Negatively, pollen grains of several species cause severe allergies. This is a negative aspect of the pollen grains, and bronchial affections. In some people, often it leads to chronic respiratory disorders such as asthma, bronchitis, etc. Parthenium weed has spread all over the country and causes pollen allergy. As I was just now telling you, the story about the hero is over and now the heroine is appearing before us. Our heroine is the gynoecium, the female reproductive organ of the plant. So this female reproductive organ is seen as a gynoecium in a flower, and it is also called as a pistil. The pistil is containing, this is the ovary, it elongates into a style and then finally it terminates as a stigma. So this is a ovary, style and stigma. The ovary is containing the placenta inside, and it is in this placenta the ovules are arranged. These ovules will be containing the egg. The structure of the ovule I am going to discuss to you in a very detailed way, then you can understand how the ovules are formed within the ovary. So ovary, style and stigma. Inside the ovary I got the placenta, and the placenta I got the ovule. Ovule is called as a megasporangium. The megasporangium is containing the integument, micropyle, chalaza, and a little more details are there in this, which I am going to explain to you by drawing a diagram directly. So please don't bother. This is also a continuation of the previous slide, how the different structures are formed inside the ovule. Now, how a ovule is there? What I am now drawing is the structure of the anatropous ovule. Anatropous ovule means a ovule which is seen in the inverted position like that. Okay, this is the one. This is called as a funiculus. The funiculus is attached to the ovule proper by in a place called hilum. The ovule itself is a oval shaped structure, that's why it's called ovule, and then it is containing two polar regions. One is called as the chalaza and another one is called as the micropyle or end. The micropyle or end is containing the micropyle. Okay. Now this is called as an outer integument and this is called as an inner integument. The outer integument is going to become the testa of the seed and the inner integument is going to become the tegmen, the inner seed coat. Okay. Now inside that you have got a tissue called a nucellus, and within this nucellus you have got what is known as an embryo sac. This is what is called as an embryo sac. Now this embryo sac is containing 8 nuclei. It is formed by the one meiotic division and then two mitotic divisions. One meiotic division, one cell by a meiotic division will give rise to 4 cells. No? Now this, the 3 will turn out to be the polar bodies. Only one will survive. This will become by mitotic division into 2, then 4 and then finally 8. Okay. So 4 will be formed here and 4 will be formed here. One from here and one from here will migrate to the center and then it will be becoming the secondary nucleus. That is why this is also sometimes called as a polar nuclei or secondary nuclei, because it is formed as a secondary product in a ovule. It's called as a secondary nucleus. Since it is migrating from a pole, you call it as a polar nuclei. Two terms are there. So 3 will remain here and 3 will remain here. The 3 which are remaining in the chalazal end here, it is called as an antipodals, and then this is called as the egg apparatus. The egg apparatus will contain one egg and then 2 synergids. Okay, so this is the structure of the ovule. Once again I repeat, the ovule is suspended or firmly held by what is known as the funiculus, hilum, and then the ovule has got 2 regions, or namely the micropylar end and the chalazal end, and in the micropylar end you have got the micropyle, outer integument, inner integument, then is less, and then you have got the 8 nucleated embryo sac. The embryo sac, 8 nucleated embryo sac, is containing 2 nuclei in the center, 3 towards the chalazal end and 3 towards the micropylar end. The 3 towards the micropylar end is called an egg apparatus, out of which 2 are called as synergids and 1 is called as an egg. The egg is going to meet the male nucleus to become the zygote, and the secondary nucleus is going to meet with another male nucleus to become the pen. What is called as a pen? Primary endosperm nucleus. Primary endosperm nucleus will become the endosperm after some time. The zygote will become the embryo. These are all the post-fertilization changes which we will be studying in a detailed way in the slides that are going to follow. Okay. So, gynoecium represents the female reproductive part of a flower. It may be monocarpellary, one pistil, or multicarpellary, more than one pistil. Latter may be free from one another, that is apocarpous condition or a syncarpous condition. What is an apocarpous condition? When the carpels are free, you call it as apocarpous. When they are fused, it is called as a syncarpous. Each pistil has a stigma landing platform for pollens. Style is elongated slender part beneath the stigma. Ovary is an enlarged portion of the pistil with an ovarian cavity. There is a locule inside it. Placenta located inside the cavity bears microsporangia. Ovule. Number of ovules varies from 1 to many. One ovule you are getting in plants such as ovate, paddy, mango, etc. And it can be many, as in the case of a papaya, watermelon, orchids, etc. See, when you just open a papaya fruit, you will be able to see so many papaya seeds, hundreds of papaya seeds will be there. Similarly, watermelon also, when you cut a watermelon for eating, you will be able to see few hundreds of seeds inside that. Orchids also contain few hundreds and thousands of seeds inside that. So, ovules may be many in some plants, and it is only one in case of certain plants. Okay. So, how a egg is, embryo sac is constituted. Just now I was telling to you this. Meiosis 1 and meiosis 2 results in, meiosis results in 4 cells, 4 nuclei. Out of that 3 are degenerating and only 1 is there. This is undergoing first mitotic division, second mitotic division, third mitotic division. 3 mitotic division leads to the constitution of 8 nuclei, which are constituted in this way, which I have just now explained it to you now very detailed way. So, see, this is a embryo sac, construction of an embryo sac. This is a funicle, where this is attached to the ovule is called as an hilum, and then this portion is called as a raphe. Then you got the chalazal end, micropylar end, nucellus, antipodals, then embryo sac. Then the embryo sac is containing secondary nucleus, egg cell, and then the synergids. This portion is called micropyle. After the fertilization, this micropyle is going to remain as micropyle in the seed also. Seed also you call it as a micropyle. Now this outer integument you are going to call as outer seed coat, then inner seed coat. This will get disintegrated. This will become after fertilization pen, primary endosperm nucleus, which will become the endosperm. 2 antipodals will disintegrate. The egg will become the zygote. Zygote will become the embryo. and the embryo will be bearing the plumule, radicle, and then the hypocotyl in between. Okay, so this is how your seed is constituted after the fertilization. See, this is only another diagram of the same thing. See, this once again repetitions, only more diagrams. So more diagrams will help you to understand the things in a very clear way. That's why I put more slides. Okay, so this is how megaspore mother cell is becoming a megaspore. 1, 2, 4, it's 4, 8, and then how it is constituted I have just shown by another diagram. That's all. Megasporangium ovule is a small structure attached to the placenta by funicle. Funicle fuses with the ovule in region of a hilum, which is in between the ovule and the funicle. Each ovule has one or two protective envelopes called integuments, outer integument and inner integument you call. Micropyle is organized where integuments are not encircled. Chalas are representing basal part, and the ovule is present opposite to the micropyle or end. Nucleus is the mass of cells enclosed within the integuments, and it has abundant reserve food, and embryo sac, a female gametophyte, is located in the nucellus. An ovule has generally one embryo sac formed from a megaspore through meiosis. So, what is a megasporogenesis? Megaspores are formed from megaspore mother cell by megasporogenesis. Ovules generally differentiate a single megaspore mother cell in micropyle or region of the nucellus. It is a large cell containing dense cytoplasm and prominent nucleus. Megaspore mother cell produces 4 haploid megaspores by meiosis. Okay. Now, what's the female gametophyte? The female gametophyte is in most part of the flowering plants. Out of the 4 megaspores, one is a functional and the other 3 degenerate. As I was telling you, one alone will be functional and 3 will get degenerated. This is what is happening in the human embryo also. 3 will degenerate and only 1 will survive. It's certain things become a rule throughout the living world, whether a plant or animal or whatever it is. The rule is a rule. There also in human being, in animals, everywhere this is a rule. The female gametophyte is 3 will degenerate and only 1 is surviving. Only one functional megaspore develops into female gametophyte, the embryo sac. The method of embryo sac formation from a megaspore is termed as a monosporic development. Monosporic development. See, these are the 4 megaspores produced. As I was telling you, these are the 4 megaspores produced. 3 will normally degenerate and only 1 will develop. Then, if an embryo sac is constituted in that way, it's called monosporic. You have got what is known as a bisporic development and a tetrasporic development also, other 3, but they are very uncommon in nature. All the types you are getting. If 2 are getting degenerated, okay, and if 2 megaspores take part in the development of the embryo sac, then it is called a bisporic development. Then in that case, what will happen? Mitosis, out of 3 mitosis, only 2 mitosis will take place. Oh, because 1 mitosis will give rise to 4, 1 more will give rise to 8. So, instead of 3 mitosis, only 2 mitosis will take place in a bisporic development of the embryo sac. Very rarely all the 4 may take part in a embryo sac development. That is called a tetrasporic development of embryo sac. Then in that case, only 1 mitosis will take place. 1 mitosis will become the 8 nuclei inside the embryo sac, and then that will be constituting the embryo sac. So, different type of embryo sac development you are getting: monosporic development, bisporic development, tetrasporic development. Monosporic development, 1 meiosis followed by 3 mitosis. Bisporic development, one meiosis followed by two mitosis. Tetrasporic development, one meiosis followed by two mitosis. So that makes the difference. Perhaps since the bisporic development of embryo sac and tetrasporic type of embryo sacs are very uncommon in nature, we don't discuss about that very much, because most of the embryo sacs are constituted by monosporic type. Good. Nucleus of the functional megaspore divides mitotically to form two nuclei which move to opposite poles. 8 nucleate stage results from two more sequential mitotic nuclear divisions which are strictly free nuclear. Free nuclear means no wall formation is taking place. There is also a controversy about that, whether each one should be called as a cell or nucleus. See what happens, one nucleus is there, it divides into two, it goes like that. Then this divides into two like this, then this divides one more like this. So all these divisions are strictly free divisions. That's what they are telling. Okay, no wall formation is taking place between the nuclei, they don't become the cell. Some people prefer to call it as a cell, 8-celled stage. That word you could have seen in some textbooks, but 8 nucleated condition is the correct word, because they are not cells, they are only nuclei. One migrates from here and one will migrate from here. So it's absolutely a free nuclear division and they don't become their cells. But cell walls are laid down, leading to the organization of the female gametophyte. Only 6 nuclei are surrounded by cell walls and organized into cells below egg apparatus. Remaining 2 nuclei, polar nuclei, are situated in the central large cell. 3 cells at micropylar end constitute egg apparatus, which consists of 2 synergids and 1 egg cell. I was telling you that in this portion is called as egg apparatus. Okay, egg apparatus. That egg apparatus is containing 1 egg and 2 synergids. This is, you have got antipodals here, then secondary nucleus here. The whole thing is called as an embryo sac. So that's what is given here. 3 cells are at micropylar and constitute egg apparatus, which consists of 2 synergids and 1 egg cell. Synergids have special cellular thickenings in micropylar tip called filiform apparatus. See, synergids have special cellular thickenings. This is very, very important. They have got thickenings and then that thickenings are called as the filiform apparatus. This is sometimes being asked in the examination. What is a filiform apparatus? Where it is seen? It is seen in the synergids or in the egg apparatus area, not in the antipodals. Filiform apparatus or filiform cells or filiform, this is known as structures. Filiform structures are seen in the egg apparatus area. Which plays an important role in guiding the polar tissues into the synergid. So these filiform tissue or filiform structures, they play a very, very important role in the structure. See, as I was just now describing to you, this is the structure of the ovule. Okay, and then you have got the embryo sac inside, egg will be here. Now what happens, the pollen tube comes and then it has to reach through this, it will come through that. So this pollen tube is able to reach this one, and the pollen tube, the tip of the pollen tube is able to recognize the egg apparatus only because of these filiform structures. The filiform structures play a very, very important role in that. The pollen tube is able to recognize that and then it is able to reach the egg apparatus successfully and very clearly, and then finally it is able to discharge the male gametes. Before that, in the topic on fertilization, we will be discussing how beautifully it is being done just before the fusion, just before the division. One male nucleus will divide into two. One will be fusing with the egg and another one will be fusing with the secondary nucleus. So the filiform structures, they play a very important role for guiding the pollen tube and then reionizing the pollen, egg apparatus, and then making the fertilization very successful. So, three cells at the chalaza end are called antipodals. A large central cell has two polar nuclei. So, this is where I left. It plays an important role in guiding polar tubes into synergy. These cells at the chalazal end are called antipodals. A large central cell has a two polar nuclei. This typical angiosperm embryo sac at maturity is 8 nucleate and 7 celled. As I was telling you, in some textbooks they are using the word cells and some textbooks they are using the word nuclei. It depends. Okay, so they say 8 nucleate and 7 celled. You don't get too much confused about these terminologies. It all depends it varies from embryologist to embryologist. Okay, fine. So what we have studied in our today's class, a very interesting important topic today we have discussed, the plant reproduction, how the plants are able to reproduce. Now the non-technical people who don't know anything about the plant side, they see the flower only as a beautiful structure. They say it's a symbol of love, it's a symbol of affection. When beauty of flower means it can be adorned to the God, it could be put on your head. It is only in that way we view the flower. But for a plant, the flower is a reproductive organ in fact. And that flower is containing the androecium and gynoecium. Androecium male sex organ, gynoecium is a female sex organ, and they are able to fuse, and then how an embryo is formed is what we have discussed in our today's class, and how this is leading to fertilization. That will be discussed separately in another lecture. Thank you very much for listening. Thank you, dear students. Thank you.